to start by giving you some background information on this trial, which is kind of unique. And I want to emphasize 3 points. First is the role of MRD assessment in ALL. The second is how to treat MRD positive patients in the best way. And the third is Blinatumomab, which is a really new treatment principle for the management of ALL. So ALL is basically a disease of the bone marrow. And traditionally, the disease is measured by simply counting the blasts on a bone marrow smear under the microscope. And the sensitivity of this method is 5%. If the blast count is below 5%, we speak of a complete hematologic remission, but we know that still blast cells are present in the body, and this is called minimal residual disease. And several methods are available in ALL to measure minimal residual disease. In our study, we focused on a PCR-based method, a quantitative PCR of individual gene rearrangement. The reason why we choose this method is that it is available or applicable in more than 90% of the ALL patients, and it is a very well standardized method for evaluation of MRD. Now, the second thing which is important here is what is the biological meaning of MRD persistence? We know from a number of clinical trials that persistent MRD leads to a very poor prognosis in ALL patients. You can see on the bottom of the slide on the right side data from the German ALL trial showing that patients with persistent MRD, which is here named molecular failure, nearly all relapse despite continued chemotherapy. So this is a group of patients which is basically resistant to conventional chemotherapy drugs. And on the left side, you can see that this high relapse rate also translates into a poor overall survival, and nowadays this is the poorest subgroup of adult ALL, which is identified by molecular failure. Now, how can we optimally treat these patients? In ALL, we usually offer patients a stem cell transplantation if they have a poor prognosis, because this is the most intensive treatment which is available nowadays. And most of the study groups have this indication now for patients with molecular failure of ALL. But unfortunately, these patients also have a higher relapse rate after the subsequent stem cell transplantation. So this is a good treatment, but still not optimal. And we need urgently new treatment approaches to treat these chemotherapy-resistant patients. And these new treatment approaches should be non-chemotherapy. And the goal is here to achieve a molecular remission, to avoid the full relapse, and also to perform then a stem cell transplantation in complete molecular remission. And we should not forget about the patients who cannot get a transplant, for example, older patients. And in these patients, of course, it's a goal to achieve a continuous complete remission with these new compounds. And here comes Blinatumomab into place. It is a new treatment principle, a bispecific antibody, which is on one side directed to CD19. CD19 is expressed on the surface of B precursor ALL blast cells. And on the other side, it is directed to CD3 and thereby attracts CD3 positive T cells, brings them in very close proximity to the target cells, and this leads to activation of the T cells. The T cells proliferate, and they perform a serial kill of the target cells, which is very effective. And there are some data from a small pilot study from Germany where we treated 20 patients with MRD positive ALL, and the response rate in this trial was 80%. And there are, of course, much more data with Blinatumomab in relapsed refractory ALL. This also led to the FDA approval just a few days ago. But the difference in this trial is that we treat the patients before the full relapse. So we included patients with MRD positive ALL above the age of 18. The detection limit had to be above 10 to the -3, so quite high MRD level, close to the full relapse. The primary endpoint of this study was complete MRD response, and of course, a secondary endpoints adverse events. There are also a number of long-term outcome parameters as secondary endpoints, but they cannot be analyzed at this time point. A longer follow-up is needed. 116 patients were included in this trial, in an international trial in several European countries. This is the treatment overview. All patients had to receive at least one cycle. One cycle means 28 days of continuous infusion of Blinatumomab. Then the primary endpoint assessment. After this first cycle, patients could receive 3 more cycles or a stem cell transplantation at any of a time point if a donor was available. Afterwards, the patient entered the long-term efficacy and survival follow-up. So this slide shows you the major result of this trial. And I would like to explain, first of all, the endpoints, because we have to get a little bit used to the new MRD-based remission assessment in ALL. So the primary endpoint was complete MRD response after cycle 1. This means no MRD detectable with a minimum sensitivity of 10 to the minus 4. There was an exploratory endpoint, which is MRD response. The difference is that also patients were calculated here who were still MRD positive but below 10 to the minus 4. And there are 2 patient populations which we analyzed. The first is the total population of all patients, and here the complete MRD response rate was 78%, and the MRD response 85%. And the second patient group where we analyzed these results is the efficacy analysis set. This is, of course, the more relevant group of patients, because these are patients who really fulfill the entry criteria, had an MRD level above 10 to the minus 3, and also no full relapse. And here, the results are slightly different: 80% complete MRD response and 85% MRD response. Interestingly, we didn't find any prognostic factor for the achievement of MRD response, so the responses were achieved in patients at any age, in patients with prior relapse, and also in patients with different MRD levels at the start of the treatment. The adverse events profile, with showing here the events with a frequency of more than 10%, has 2 types of adverse events. The first type are events which are somehow correlated to the cytokine release, like fever, chills, and fatigue. This was observed in most of the patients, but in most cases the adverse events were only grade 1 and 2. And the other type of adverse events are neurologic events. Most frequent symptoms were tremor and aphasia in 29% and 13% of the patients. And also here, most of the events were grade 1 or 2. But still, these events are clinically relevant because they can lead to treatment interruptions in some of the patients. This is, of course, something which we do not like to do in these patients. To conclude, this is the first international multi-center trial in adult ALL with an MRD-based patient inclusion and MRD endpoint established by central MRD assessment in a central lab. Most patients achieved a complete MRD response within 1 cycle, so a very rapid response. Responses occurred in all subgroups of the patients. Adverse events were mostly related to the T-cell activation, but there were also clinically relevant neurologic events. Most of them had, although grade 1 or 2. The follow-up data of this trial will show whether the good MRD response rate will translate in a long-term outcome benefit. For me personally, this trial is very important because it's an up-to-date trial where we use new methods for PCR-based methods to identify patients with a high risk of relapse and treat them before the relapse occurs. The second point is that we use a new endpoint, which is of course also MRD-based. And the third is that we use a new non-chemotherapy treatment to eradicate this highly resistant, persistent ALL subgroup.